For this project situation, we had the help of the Pontifical Catholic University of Chile, especially from the Faculty of Agronomy and Natural Systems, as they conducted an 8-week experiment on lettuce and cabbages. For each type of land, they had 4 different groups. Each group had 5 pots, and each pot had 3 plants. In each group, they had different type of mulch. The first group was a control group, the second group had shredded newspaper, the third group had wood shavings, and the last group had black plastic. In the next weeks, they measured the height of each plant and the percentage of undergrowth that there was in each pot. The null hypothesis for this linear regression is that there is no relationship whatsoever between the percentage of undergrowth and the plant growth. However, the other hypothesis is that there is a relationship between the percentage of undergrowth and the plant growth. Additionally, they weighted the biomass at the end of the last week. To do the statistical analysis, we're going to use Minitab. Firstly, we did a linear regression. This where the percentage of the undergrowth is going to be the predictive variable, and the height of each plant is going to be the response variable. This will allow the identification of any potential relationship between the undergrowth and the plant growth, as well that assess whether plant growth can be predicted from the percentage of the undergrowth. We determined that linear regression is insufficient to determine the effectiveness of the Mulch methods. Because of this, we're going to do different ANOVA tests. The null hypothesis for the ANOVA test is that the mean plant growth is the same for all undergrowth control methods. And the other hypothesis is that at least one method produces a different mean plant growth. Also, the biomass measurements will be analyzed to provide an additional indicator of the plant development.
Now let's go with the linear regressions. First of all, we have the figure number 1, which represents the regression analysis between the percentage of undergrowth and lettuce growth. Its p-value is of 0.172. and its R-square is of 3.19%. Then we have figure number 2, which represents the residual plots for the linear regression of cabbage versus undergrowth percentage. Its p-value is lower than 0.05, specifically 0.005, and its R-square, its coefficient of determination, is of 12.99%. Then we have the technique versus growth figures in the ANOVA's tables. We have figure number 3, which represents the interval plot comparing the mean growth of lettuce among 4 mulch treatments. Then we have in figure 4 that although differences among the treatment means can be observed, since we analyze different treatments, the confidence intervals overlap substantially, which indicates that these differences may not be statistically meaningful. And then we have figure number 5, which displays the effect of mulch technique on cabbage growth. We have then the figure number 6, showing the biomass produced which varied among the different mulch treatments. And well, finally we have figure number 7, which is the technique versus undergrowth percentage, and it illustrates the relationship between mulch treatment and undergrowth percentage. A clear difference among treatments can be observed. For example, the plastic produced a lower percentage of undergrowth. And that would be it for my part.
The results suggest that mulch type influence both weed development and crop performance. Different mulch materials create different soil conditions, which can affect water availability, temperature, and sunlight. The weed control results show that black plastic mulch was more effective at reducing weed growth than wood mulch. This may be because black plastic blocks sunlight, making it more difficult for weed seeds to germinate. The relationship between weed growth and crop growth was different for each species. For lettuce, the regression analysis showed not significant relationship between weed growth and crop growth, with a p-value of 0.172. This suggests that weed presence was not the main factor affecting lettuce growth during the study period. Other environmental factors may have had a greater influence. For cabbage, a significant relationship was found between weed percentage and plant growth, with a p-value of 0.005. This indicates that cabbage may be more sensitive for competition for weeds than lettuce. However, R-square value was relatively low, meaning that weed growth alone does not explain difference observed in crop growth. In conclusion, results show that mulch can be a tool very effective for weed management, but its effect on crop performance depends on the crop species and other environmental conditions.